Hi all, in this video we will be speaking about the top most used equipments in food laboratory like weighing balance, protein analyzer, HPLC, burette, um, magnetic stirrer and etc. Hopefully you will gain knowledge about all the types, features and application of all these equipments. The first one is digital balance. So digital balance is used to measure the substances with high accuracy. There are different types of digital balance like analytical balance, precision balance, micro balance, ultra micro balance. In this one you are seeing the normal basically used one in laboratories. So this can be used to digitally display accurate readings of the weight. Tearing function is to exclude container weight and other vessels weight. So for the application test, weighing sample preparation, weighing chemicals, standard preparation, culture medium preparation, buffer preparation. So the second one is protein analyzer. So this can be used to measure proteins in different dals and other protein rich foods like milk and etc. So this determines the protein content in food, feed, pharmaceutical products, biological samples and etc. The most used protein analyzation method is schedule dal method. This thus has an distillation unit, digestion unit and titration unit. So in digestion unit the food particles is placed and where the amino acids is removed and it is secreted into an HClC acid, and in distillation unit it is removed and measured in titration unit according to the color change amount of nitrogen present in the food sample is measured. So it has an automated sample processing in modern equipments and it also high throughput capability. Advantages are it provides high accurate nutritional analysis essential for food labeling and other researchers. In food industry it is also used in protein content analysis in milk, meat, cereals and also in animal feed regularization and quality control. So in distillation unit you can see that from digestion unit the liquids will be secreted into distillation unit. So application is basically on protein analysis, schedule dal method, digestion of sample, distillation of solution. So third one is pH meter. So as we all know, pH is essential one in pH meter. So the third one is pH meter. As you all know, pH is the main important function in food industry because it maintains the amount of hydrogen ions in the food packagings, material and food products. So according to this, the pH measures the hydrogen ion concentration of a solution, indicating how acidic or basic it is. So high acidic foods like pickles and vinegar and it all needs a regulation over pH. So using this pH meter which can be so useful to detect the pH. The pH of water is used as another solution. Acidity check, alkalinity check is also other applications of pH meter.
Next we are going to see about the burette. A burette is a long graduated glass tube with a stopcock at the bottom used to deliver precious volume of liquid, especially used in the titrations. Glass burette. Mostly the burette made up of two materials. One is glass, another one is plastic. This glass burette are also used mainly in the chemistry experiments. A burette is a long calibrated glass tube, usually 50 ml capacity, stopcock for controlled liquid release, high accuracy in measuring delivered volume, can be glass or plastic. It is also used for the acid-base titration, redox and complexometric titrations, measuring and delivering precious volumes of solution, and it is also used for the chemistry analytical experiments. Applications: titration for analytical purpose, measurements of variable amount of liquid used as a titrator, etc. Next, we are going to see about the HPLC. HPLC stands for High Performance Liquid Chromatography. High performance liquid chromatography is an advanced analytical technique used to separate, identify, and quantify components in a liquid mixture. It works by passing the sample through a column packed with a stationary phase under high pressure, while a liquid mobile phase carries the analytes at different speeds based on their chemical interactions. Features of HPLC are high pressure system, use pump to push solvent mobile phase through the column at the pressure up to 6000 psi. Column packed with the stationary phase, may be in silica or polymer based materials. Detector UV fluorescence or mass spectrometer or refractive index detector for measuring separated compounds. Automation. Modern HPLC systems are computer controlled for precision and reproducibility. Versatility. Can separate both the organic and inorganic polar and non-polar molecules. Accuracy and sensitivity. Detects compounds even at a very low concentration, ppm to ppb range. Types: normal phase, reverse phase, ion exchange, size exclusion, and affinity HPLC. Uses of HPLC. This HPLC mainly used in the pharmaceutical industry for drug purity testing, stability, and formulation studies. And also it is used in the food and beverage industry for analysis of vitamins, preservatives, sweeteners, caffeine, etc. And it is used in the clinical and biomedical research for detection of hormones, proteins, amino acids, and metabolites. It is also used in the environmental studies measuring pesticides, herbicides, and pollutants in water, soil. Next, we are going to see about the separating funnel. The definition of separating funnel is a laboratory glassware used for separating immiscible liquids based on their density difference. It is pear-shaped or cylindrical with a stopcock at the bottom, allowing controlled release of liquids. The features of the separating funnel are: first, the shape. Typically pear-shaped with a conical bottom at a cylindrical neck. Stopcock. Teflon or glass stopcock for controlled drainage of liquid. Capacity available in various sizes like 50 ml to several liters.
Hot plate with magnetic stirrer. It is a small magnetic stir bar. It is placed inside the liquid container. The hot plate generates a rotating magnetic field using an inbuilt motor or electromagnet, causing the stir bar to spin and mix the solution uniformly while heating. It is used to heat solutions and simultaneously stir them with the help of a magnetic stir bar. It dissolves solids into solvents, preparing buffer solution, and it maintains a uniform temperature in chemical reactions. It is useful in microbiology, chemistry, food technology, and pharmaceuticals. It provides both heating and stirring at the same time, ensuring homogeneous mixing and preventing localized overheating. The main components are hot plate surface, usually made of ceramic, aluminum, or stainless steel, resistant to chemicals and high heat. Magnetic stirrer mechanism. It contains a rotating magnet or electromagnet beneath the surface. Magnetic stir bar. A small Teflon coated bar placed inside the container with the liquid. It rotates due to the magnetic field. Control panel. It separates the knob or digital control to adjust temperature and stirring speed. Heating element. It provides uniform heating. The applications are chemistry and biochemistry labs. Heating or boiling a solution or liquid, mixing a solution or liquid, heat and stir a solution or liquid simultaneously. And also pharmaceuticals and microbiology. The next laboratory equipment is lactometer. It is a simple instrument used to measure the purity of milk by determining its specific gravity. It is a type of hydrometer since milk contains water, fats, proteins, and other solids. The lactometer floats at a particular level depending on the density. If extra water is added, the density decreases and the lactometer sinks deeper. It detects the adulteration of milk with water and it controls the quality in dairy industry, and it is used in laboratories for routine milk testing. The advantages are low cost, easy to use, provides quick result. Lactometer application test measures the milk density, purity testing of milk, and measures content of water in milk. Next laboratory equipment is blender or mixer. Blender. It is used to mechanically mix, crush or emulsify food and other substances. The working principle of blender is it consists of sharp rotating blades powered by a motor at the base. The high speed rotation creates a vortex that pulls ingredients down toward the blades, breaking them into smaller particles and ensuring uniform mixing. The applications are food industry, it preparing purees, sauces, smoothies. Laboratory, homogenizing samples, preparing test solutions, blending small plant or animal tissues for analysis. Advantages: quick homogenization, versatile, easy to operate, and clean. The application test. It grinds semi-solid samples, chemicals or ingredients. It blends different samples, chemicals or liquids. It mixes and crush off solid. The next laboratory equipment is fume hood, a ventilated enclosure used in laboratories to safely carry out experiments involving hazardous or volatile chemicals. The working principle: the fume hood pulls air from the lab and expels it outside through a ventilation system, preventing harmful vapors, gases, or dust from spreading into the lab. A glass sash provides a barrier between the user and the chemicals. Applications: handling toxic, flammable, or corrosive substances, protecting the lab personnel from chemical inhalation and splashes. Application test: working with different hazardous or odorous chemicals, releasing toxic and hazardous fumes outside, working in closed chamber, ensuring safety.